My name is Ken Cho. I'm from University of California, Irvine. And it is my pleasure to introduce this year's recipient of the Elizabeth D. Hay Award. And this award is given to the individual who has made a significant impact early in their careers, and therefore capturing the spirit of Dr. Betty Hay and her inspiring educational contribution. And there are many undergraduates and graduate students who do not know about Dr. Elizabeth Hay. Just let me spend a couple of minutes to introduce her contribution and then today's main speaker. And Dr. Hay had a distinguished career in science, and she was best known for her research in limb regeneration and the role of extracellular matrix in cell differentiation, and then also epithelial mesenchymal transitions. And one of her key studies involved understanding how a salamander can regenerate it after an amputating limb. So what she found was that after amputation, she discovered that the differentiated cell could be de-differentiated and then revert back to being in unspecified cell types. And this discovery closely parallels the principle of modern developmental biology that we study today. But she did this over decades ago. Now, in addition, Dr. Hay demonstrated that epithelial cells produce extracellular matrix proteins in various organ systems, and she found that these proteins instruct other cells about the specific cell type they should develop into. Now, Dr. Hay made history in 1973. She became the first woman to be elected as the president of the Society for Developmental Biology. And she continued her pioneering journey as the first female recipient of the SDB Conklin Medal, and later the president of the American Society of Cell Biology. And in 1984, she achieved another milestone, and she was elected to become a member of the prestigious National Academy of Sciences. Now, we turn our attention to the individual who has embodied Dr. Hay's pioneering spirit in our present day, Dr. Aimee Jaramillo-Lambert. And she is currently a tenure-track assistant professor in the Department of Biological Science at the University of Delaware. And she has dedicated her work to elucidate the mechanism of how each egg and sperm acquires the correct number of chromosomes during meiosis and how chromosome shape is determined in C. elegans. Dr. Jaramillo-Lambert attained her PhD degree from the University of California, Davis, where she ventured into uncharted territories by pioneering the lab's first C. elegans project under the mentorship of Dr. Joanne Engelbrecht. And her groundbreaking thesis on sex-specific differences in the C. elegans germline has since become a cornerstone of her research. Dr. Jaramillo-Lambert continued her work with C. elegans during her postdoctoral period, this time at the NIH with Dr. Andy Golden. There she uncovered the role of topoisomerase II in meiosis, identifying paternal-specific alleles of topoisomerase II in C. elegans, and then when she examined the effect, the mutation had no effect during oogenesis. This finding revealed that even though meiosis is highly conserved, the process can be significantly different within the same species, between males and females. This work provided a rare glimpse into the paternal inheritance and novel sex-specific effect of topoisomerase. And since she became an independent assistant professor, she has diligently continued this work in her lab, and she recently completed detailed mechanistic study that elucidates the unique role of topoisomerase II in maintaining chromosome cohesion during spermatogenesis. This work has deepened our understanding of the distinct developmental specialization between male and female gametes, and her future plan involves continuing continuing with this project. Lastly, Dr. Jaramillo-Lambert's research achievement only tell a part of her story. She is also a passionate educator, keen on guiding her students and nurturing their curiosity, and she is an advocate for equity and inclusion. She is indeed an embodiment of the spirit Dr. Hay herself represented, and I invite you all to join me extending our heartfelt congratulations, applause to Dr. Aimee Jaramillo-Lambert.
I would like to thank the Society for this award, and special thanks to my colleagues that nominated me. It's their continued cheerleading and support that has pushed me in this field and to keep going, and I truly appreciate them. I'd also like to acknowledge that my journey has not been in a vacuum, and I am lucky enough to have amazing friends and colleagues that continue to give me support and advice throughout my scientific career. And I've also had truly amazing mentors, both in graduate school, where I worked with Dr. Joanne Engebrecht, and during my postdoctoral training, where I worked with Dr. Andy Golden. I've also had the privilege of working with a group of amazing undergraduates and graduate students, postdoctoral researchers, and lab technicians. It's their hard work that has led to all of this research and keeps the lab going. So my lab broadly studies sexual reproduction. During sexual reproduction, diploid organisms have a set of cells that are set aside during development that go through a specialized cell division called meiosis to generate gametes, or cells that have half the number of chromosomes as the adults. And during fertilization, these gametes will come together and restore the chromosome number, and then the development of a new embryo can begin. And within this process of sexual reproduction, we focus on two main questions. First, we're interested in understanding the mechanisms that contribute to differences in meiosis between the sexes, specifically looking at chromosome structural differences. And the second question that we are trying to address is determining whether there are other components or proteins that are provided by sperm that are needed for early embryonic development. The main focus of sperm has mainly been in the haploid DNA or genome and the paracentrioles that it provides for the first mitotic divisions, but there's evidence that there's other proteins that they provide and RNAs that are needed for early development. And for the purposes of this talk today, I will focus on this first question, looking at meiosis. So meiosis is the specialized cell division that allows diploid organisms to generate haploid gametes for sexual reproduction. And during meiosis, chromosomes must first replicate their DNA, and then homologous chromosomes must pair and align along their lengths, and this culminates in the formation of a proteinaceous structure called the synaptonemal complex being laid down between the homologous chromosomes. And within the context of the synaptonemal complex, crossover recombination occurs, forming physical links between the homologs that is essential for their segregation at the first meiotic division. Following crossover formation, the SC disassembles, and then there's a chromosome remodeling and restructuring event that occurs in late meiotic prophase, where the chromosomes have to condense and they take on a new shape, where we usually think of this as a cruciform shape with a short arm and a long arm. And this structure is essential for the separation of the homologous chromosomes at the first meiotic division, and this is followed by a second division where the sister chromatids separate to form the haploid gametes. So to study meiosis, my lab uses the awesome model organism, C. elegans, and this slide is showing the C. elegans reproductive system. To study sex-specific differences, it's great that C. elegans exists as two sexes, hermaphrodites and males. Hermaphrodites produce both sperm and oocytes, but they produce sperm during the larval development, and they store them in a special compartment called the spermatheca. And then when they become adults, the germline switches to only making oocytes. So C. elegans adults are essentially females with stored sperm. Males, on the other hand, only produce sperm throughout their lifetime. So C. elegans germline is housed by the syncytial gonad. The hermaphrodite has two U-shaped gonads that converge on a central uterus. And what's really great about using this system for studying meiosis is the way the germline is arranged within the gonad. So if we dissect a gonad out of the worm and stain it with DAPI, we can visualize all the stages of meiosis based on chromosome morphology. So at the distalmost end of the gonad is a population of mitotically dividing cells that serve as the stem cell population for the germline. As the germ cells progress down the gonad tube, they enter meiosis in the transition zone, and then they undergo an extended meiotic prophase where we can visualize all the different substages of meiosis. And in this hermaphrodite gonad and the most proximal area, there's a chromosome condensation zone where the chromosomes are condensing and remodeling as they prepare for the homologous chromosomes. And in hermaphrodites, the homologous chromosomes condense and individualize such that we can observe all 6 homologous chromosome pairs. The male germline has the same spatial temporal arrangement of the gonad. They don't have, oh, sorry, I forgot to say that in hermaphrodite germline, they undergo a pause before they get fertilized, so they pause in diakinesis, and then after fertilization, the meiotic divisions resume. The males don't have this pause, and they can go straight into the meiotic divisions. But I want to point out there's another difference between the male and the hermaphrodite germline, and that's in this condensation zone where the males have an extra step of meiotic prophase, and here the chromosomes condense, but instead of individualizing like they do in the hermaphrodite germline, they coalesce into the spherical ball of chromatin. And then after this stage, they can enter diakinesis and continue with the meiotic divisions to form haploid sperm. So this whole project started with the identification of a novel allele of topoisomerase 2. So topoisomerase is a highly conserved enzyme from bacteria to humans. It's essential for disentangling chromosomes and solving DNA supercoils. But this particular allele, which we call IT7, and it's an arginine 828 to cysteine change in the catalytic domain of this enzyme, is a temperature-sensitive mutation, such that at the permissive temperature, homozygous mutants have a wild-type phenotype, but at the restrictive temperature, homozygous mutants show a mutant phenotype. And what was particularly interesting about this allele is that it had a sex-specific phenotype, such that when we raised these animals at the restrictive temperature, spermatogenesis had chromosome segregation defects. So on the left is a picture of a wild-type germ cell that's undergoing spermatogenesis, and the haploid complements of DNA, shown in red, have been segregated into the budding spermatids. In the top 2 IT7 mutant, the chromosomes fail to segregate at meiosis 1, and they don't segregate into the budding spermatids. So these animals produce sperm without DNA. They can still fertilize, but the resulting embryos die because they're haploid. So we saw these segregation defects in 100% of the sperm that we observed, but no chromosome segregation defects were observed during oogenesis. And topoisomerase is expressed during oogenesis, and it is essential for oogenesis. So our big question at this point was, why is the spermatogenic germline more sensitive to loss of top 2 function? And this work has been carried out by a graduate student in the lab named Christine Rourke. So I want to remind you that in the C. elegans germline, between the sexes, there's a difference in the chromosome condensation zone at the proximal end of the gonad, where the chromosome morphology differs significantly between the sexes. So Christine started this project by looking to see whether different chromosomal structural components might be altered in the top 2 mutant. And she started this project by looking at the localization of the meiosis-specific subunit of cohesin called REC8 in spermatogenesis. So I have a cartoon on the upper right just showing you where in the gonad we are looking. And so in wild-type germlines during spermatogenesis, REC8 localizes to what's called the long arm of the chromosomes, and it often looks like, to me, like a spokes on a wheel. And then REC8 still associates with chromosomes as they segregate at anaphase 1. In the top 2 mutant, she saw that REC8 could still localize to chromosomes in metaphase 1. However, REC8 was absent from the chromosomes in anaphase 1s, suggesting that top 2 might be needed to maintain REC8 on the chromosomes. So the pathway for regulation of REC8 removal had been previously worked out in C. elegans oogenesis. And the labs that studied this found that AIR2, the Aurora B kinase, is required to phosphorylate REC8 specifically on this short arm of the chromosomes, which marks it for removal to allow for the homologous chromosomes to separate. So we saw that REC8 was mislocalized, so she looked at AIR2 localization. So in wild-type animals, AIR2 localizes to the short arm of the chromosomes and associates at a distinct location on the anaphase 1 chromosomes as they separate. In the top 2 mutant, AIR2 localized to the metaphase chromosomes, but in the anaphase 1 chromosomes, AIR2 was ectopically localized where it's not restricted to specific domains of the chromatin and instead appears to be located all over the chromatin, suggesting that this could be the reason why that all of AIR2 is getting removed instead of just a distinct subset of REC8. So remember that we are interested in sex-specific differences, and even though we didn't identify any chromosome segregation defects in the top 2 mutant, we still wanted to look at the localization of these components in both wild-type and top 2 mutants in the female germline. And just as a summary, I want to tell you that, again, remind you that we found that in spermatogenesis, AIR2 is ectopically localized and REC8 is prematurely removed. And for the sake of time, I will tell you that in oogenesis, REC8 localization looked wild type, which is what we thought we would see. But then we looked at AIR2, and here we're looking at the most proximal oocytes, so where the chromosomes are most condensed in diakinesis and take on these cross-shaped structures. And then we're also looking at the fertilized oocyte after, where it's in prometaphase, so these occur after fertilization. So in wild-type oogenesis, AIR2 localizes to the short arms of the chromosomes, and it's also on the short arms of the chromosomes in prometaphase 1. But when we looked in the top 2 mutant, to our surprise, AIR2 was not localized on the chromosomes in the -1 oocyte, but it was there on the prometaphase chromosome. So top 2 seems to be regulating the localization of AIR2 in some way. So because of the delayed localization of AIR2 in top 2 IT7, we wanted to see if the top 2 does indeed play a role in the timing of AIR2 localization. And so far I've told you how AIR2 is spatially regulated in oogenesis, but AIR2 also has a second role, or regulatory role in oogenesis. And this is a temporal recruitment, which is regulated by the maturation promoting factor. And so Christine asked if she depleted WEE1.3 in the germline, could we now rescue localization of AIR2 in the top 2 mutant? So Christine performed control RNAi and WEE1.3 RNAi in the top 2 IT7 line, and in the control, she saw that, just like we saw previously, that there's no AIR2 localization in the top 2 mutant in the -1 oocyte. But when she depletes WEE1.3, AIR2 localization is restored, suggesting that top 2 may be playing a role in the timing of regulation in oogenesis. So meiosis is a very complex process that requires a lot of different proteins and genes to accurately segregate homologous chromosomes and then sister chromatids. And topoisomerase 2 has been extensively studied in mitosis, but how it interacts with the meiotic machinery had not been previously known. So we next asked, where does top 2 fit in the meiotic pathway? And this project has been largely spearheaded by a recently graduated PhD student, Dr. Narajan Bandari, but it has also been the work of several other people in the lab. And so to get at this question, we used the awesome power of C. elegans genetics, and we performed a genetic suppressor screen. The top 2 animals produced dead embryos because the sperm don't have DNA, and so we mutagenized this line and then looked for animals that have a second mutation that somehow fix the original top 2 defect, and now we have viable progeny. And with this genetic suppressor screen, we identified 11 suppressors of top 2 IT7 embryonic lethality. And these are all missense mutations in various different genes. But for today's purposes, I'm going to look or tell you a little bit about one particular suppressor, and that's these 7 different suppressor mutations that we found in one gene called TDPT1. So TDPT1 is the C. elegans homolog of human tyrosyl DNA phosphodiesterase 2, or TDP2. And so this enzyme is well known to interact with TOP2 during mitosis. So TOP2 normally is a relatively fast enzyme, and to solve DNA supercoils or DNA entanglements, it binds to DNA as a homodimer, creates a double-strand break in the DNA. A second strand of DNA is allowed to pass through that break, and then topoisomerase 2 will reseal the break. But sometimes TOP2 gets stuck on DNA, and this is toxic to cells. So if this happens, TDP2 recognizes TOP2 DNA complexes and will come in and remove TOP2 from the DNA, now providing a substrate that can be repaired by either non-homologous end joining or homologous recombination. So the first question we asked is, how do the different mutations affect TDPT1 function? So we mapped these different suppressing mutations to the TDPT1 crystal structure, which had been solved for C. elegans, and they all map to the catalytic domain of the enzyme, which is shown here. This is only the catalytic domain. But we figured that these various mutations could affect the protein in several ways. It could change the localization of the protein, could impair its catalytic activity, or maybe it just destabilizes the protein. So we started by creating a worm scarlet tagged TDPT1 to look at localization of TDPT1 in the germline. And TDPT1 localizes in the nucleoplasm of germ cells, and it's found in all germ cells from the distal tip to the proximal gonad. And then we used CRISPR-Cas9 genome editing to recreate one of the best suppressors, which was glycine at amino acid position 270 to aspartate in the worm scarlet TDPT1 background. And we found that TDPT1 was not expressed in the germline, or at least we couldn't detect it. And this was confirmed when we did Western blots, so the TDPT1 mutant, the TDPT1 protein is greatly reduced. And then Narajan spent a lot of time expressing the TDPT protein in E. coli cells and also creating all the different mutations and expressing them in E. coli, and looked at the thermostability of the proteins, and we found that all 7 of the suppressors destabilize the protein, such that they're probably just not expressed at all. So here's the wild type with a melting temperature of 45 degrees, and all of the suppressors are shifted lower melting temperature. So what do we think is happening in regards to TOP2 and TDPT1? Well, normally, TOP2 is a fast enzyme that we think binds to the DNA during meiosis, and its activity is required for chromosome remodeling and restructuring in late meiotic prophase. And this chromosome remodeling and restructuring allows for accurate chromosome segregation. In the TOP2 IT7 mutation, we think the enzyme is slow. It is not completely absent. And in this case, TDPT1, when it's around, thinks that TOP2 is stuck on the DNA. And so it comes in and removes TOP2 before it can finish its chromosome remodeling, and we get chromosome segregation defects. But if TDPT1 is absent, TOP2 can keep chugging along and do its activity and allow for accurate chromosome segregation. So the last part of the talk, I want to get into some of the newer projects in the lab. And this is looking at factors that determine the sex-specific chromosome morphology in late meiotic prophase. And so we know that these chromosomes have different shapes, and I'll show that to you in the next slide, but how is this achieved? And this project has been the work of a former postdoctoral researcher in the lab, Lauren Salviti, and a recently graduated master's student, Nicole Fung. So in many organisms, it's been observed that chromosomes of one sex are a lot smaller than the other. Generally, it's the sperm. And so we were interested in this in C. elegans, but the actual chromosome lengths between sperm and oocytes had never been measured before. And so we focused specifically on the most condensed state of chromosomes in meiosis, and that's the diakinesis stage of meiotic prophase 1. And these are a picture of an oocyte and a spermatocyte, and they're at the same scale. You can see it's pretty obvious that the sperm chromosomes are much smaller, but we did go ahead and measure them. And indeed, the average size of chromosomes in spermatogenesis are significantly shorter than in oogenesis. But what is causing this difference? And we think that TOP2 might be one of the proteins that is involved in this protein, but are there other proteins involved in this process? It's probably not TOP2 by itself that is contributing to this size difference. So we took a targeted mutant approach to this to start with. And Lauren looked at components that were known to play a structural role in chromosomes to look at there was different effects when we looked in males and females germlines. And she looked at mutants in 2 histone methyltransferases, MET2 and SET25, and a sperm-specific DNA binding protein called HTAS1. And what Lauren found was that when we took deletion mutants of MET2 and HTAS1, the chromosomes in spermatogenesis were significantly decreased, but there was no significant change in these mutants in the oocyte. And conversely, the SET25 mutant had more of a significant change in chromosome length in oogenesis than it did in spermatogenesis. So after Lauren did this, Nicole took over this project, and she focused on the histone methyltransferases. So there's a difference in chromosome length in these mutants, but does this mean anything? Does it have an effect? So to start getting at this question, Nicole looked at brood sizes of these mutants, and she found that in MET2 animals that there was a significant reduction in brood size, but there was no significant change in brood size for SET25, suggesting that it's not a direct cause, a correlation that MET2 differences in chromosome length could be affecting fertility. So how could this change in chromosome size be causing a defect in sperm quality? So MET2 is a histone methyltransferase that adds mono and dimethyl marks to the histone H3, which normally is correlated with transcriptional silencing. So to see whether there's a change in the global transcription in spermatogenesis, Nicole did some immunostaining to look at the localization pattern of the active form of RNA polymerase II. And the pictures I'll show you are looking here in the division zone. So in wild-type animals, it's been previously shown, and Nicole recapitulated this, that RNA polymerase II, the active form, is localized throughout the germline, but it abruptly turns off at the condensation zone, where the germ cells enter the karyosome. In the MET2 mutant, Nicole found that RNA polymerase II eventually does come off, but there's an extension of RNA polymerase II staining into the condensation zone, which I think it's hard to see here, but the arrowheads are pointing to germ cells that still have RNA pol II staining. So now it looks like that maybe this change in MET2 and chromosome condensation might be having effect on global transcription in the male germline. So this is taking me back to the title, that sexually dimorphic meiotic programs create specialized gametes. So we found that TOP2 is localizes to the chromosomes, and it has a different effect on chromosome structure in the male and female germlines. And we're recently discovered that these two histone methyltransferase also seem to have effects on chromosome structure in the germline. And there's probably more components that are involved. And we still don't even know what these things are doing yet. So we still have a lot of work left to do that I'm very excited about. And with that, I would like to thank my lab members, past and present, collaborators and funders, all of you for listening today. And I just want to say one last thing. So at the beginning, I mentioned my postdoctoral advisor, Andy Golden. He recently passed away earlier this month. He was an amazing person. He's the person that introduced me to the Society for Developmental Biology. Like I said, he was an amazing mentor during my postdoctoral career, and he was still mentoring me through my transition as a junior faculty, and he's just dearly missed. So thank you all,